third presentation this morning in this first session is by Dr. Brandon Boor. Brandon earned his Ph.D. at the University of Texas at Austin. He has research experience from Finland and at NIST locally, and he's recently assumed a position as assistant professor at Purdue University. And I can't help but mention that Rich Corsi just jotted on my bio, I'm really proud of Dr. Boor. So,
okay, thank you, Rich. Can you hear me with this microphone? Okay, so I will talk about particle resuspension.
Wear the mic, and you're going to look at the screen. You have to put it on your other lapel, otherwise your face is aimed away from it.
Okay,
Brilliant.
Sorry about that.
Yeah.
So I like that all of you envision yourself as being surrounded by a cloud of particles, just like Pigpen here from the Peanuts comics. This is actually a quote from Pigpen. You know what I am? I'm a dust magnet. I think we're all dust magnets. Maybe some of us are stronger magnets than others. But the fundamental process that's creating this cloud of particles is particle resuspension. And this is a process by which deposited particles are detached from a surface when they are exposed to various removal forces such as aerodynamic lift and drag, surface vibration forces, electrostatic forces. They detach and they become airborne. So we know that this particle resuspension, human-induced particle resuspension, is associated with various activity patterns and different types of movements. Of course, just walking across a carpet will resuspend particles. We know that its concentrations of resuspended particles are linked to occupancy, number of people in an occupied room, such as this. We certainly know a lot about walking, but comparatively less about resuspension of crawling infants. So infants crawl and play, and they're very close to the floor, so a source proximity effect there. You can see here. We also move when we're in bed. This might stir up mattress dust, for example. So this was a plot looking at PM 2.5, PM 10 emission rates for different indoor sources by a literature review on indoor particle resuspension. And we can see from this figure that particle resuspension is an important indoor source of particles. Emission rates for PM 2.5, for example, associated with walking-induced resuspension can range from 1 to 10 milligrams per minute. And to put that in perspective, I estimated, based on just some typical activity patterns, that this would correspond to 10 to 100 kilograms of dust that you may resuspend over your lifetime. So that's a significant mass of dust that's being aerosolized just due to walking or other movements. We know that resuspension is a source mechanism for all the stuff that's in-house dust. Of course, a lot of this is of biological origin. We have human and animal-associated bacteria. We have fungal spores and their fragments, pollen grains. Of course, we have allergens, as Lynn discussed. And we also have various chemical contaminants. So house dust is an accumulation zone and a reservoir for many semi-volatile organic compounds, phthalates, organophosphates, and so forth. You can see from these SEM images that these particles, especially biological particles, are very diverse in their shape and their morphology. This is something very interesting. In addition to just looking at resuspension as a source of particles, we also have to consider our exposure to resuspended particles and the associated biological material or chemical contaminants that may be carried on these particles. One thing I think we certainly need more focus on is infants and what we could call the infant near floor microenvironment, because these infants are very, very close to the carpet. They play, they do all different types of activities. It's very, very complex. Then we have walking. Of course, we can be exposed to particles that we resuspend from the carpet ourselves. But in an occupied environment, you may be exposed to particles that somebody else resuspends as they walk by. And then the sleep microenvironment, another microenvironment, something I think we know generally very, very little about, just in indoor air quality in general. We spend one-third of our lives sleeping. And of course, when we're in bed, we move. We have different types of movements. And this may induce resuspension of mattress dust. Mattress dust is also a reservoir for a lot of biological material. And then we have occupational workplace exposures. We may have like worker type outfits or clean room suits that may become contaminated over time with various types of particles, and then those may be resuspended. So here's just a timeline of some of the, what I would, in my opinion, some of the seminal studies related to particle resuspension, some indoors, some outdoors related to wind-blown dust. Some of the pioneering work in this area was done by Lynn and Andrea Farrow. And Andrea and her group at Clarkson have done some of the most comprehensive work on walking-induced particle resuspension using mechanical foot as well as full-scale chamber measurements. So this is really has framed the body of knowledge that we have. We have learned a lot in the past few decades, but there's certainly a lot to learn moving forward. And the studies that I just showed and others, we use a material balance model, as some of you may know, to mechanistically evaluate particle resuspension. So we can treat resuspension as a source, look at the concentration in the air, and also the surface concentration, which would be L. And this is something I think particularly interesting in terms of the size distribution of dust, which I'll describe in a few slides. But the source term for resuspension, we can classify with a few different metrics, one of which would just be a bulk emission rate of particles released per hour. Then we also have a resuspension rate and a resuspension fraction. These two, I think, are preferred because they're linked to the source. They're linked to the surface concentration and size distribution of dust particles. The resuspension fraction, as it's defined here, is also linked to the movement frequency. So this is useful for the varying types of activities and movements and their frequency, such as footfalls and so forth. So moving forward with the rest of my talk, I would like to structure particle resuspension around these three elements. First of which is dust. Looking at dust, the physical characterization of dust, composition of dust particles, their adhesion to the surfaces, indoor surfaces, and each other. Then we have the resuspension process itself, whereby these settled particles become airborne. They detach from a surface. And then we have the ultimate transport to the breathing zone where we're ultimately exposed to these resuspended particles. So there's three elements here. The first I'll talk about is dust. So I think there's quite many epidemiological studies where dust from either flooring or mattresses is collected and then analyzed for content of SVOCs or microorganisms. So we have a large body of data, large database of dust loads and mass fractions of SVOCs and so forth. You can see here some ranges of dust loads. It varies quite a bit in indoor environments. We may have very light dust loads. For example, on a mattress, less than 0.1 grams per square meter. That's grams of dust per square meter of mattress. And then in ventilation ducts or really dirty carpet, we may have dust loads in excess of 10 grams per square meter. Now, although we have a lot of data on dust loads and information on stuff in dust, we have very little data on the size distribution of dust particles. And I think this is something that's really a future research need. Here's an example, just an illustration of a volume or mass fraction of dust. This can be done with various techniques, laser diffraction using an instrument called a mastersizer or microscopy. But getting information about how these particles, their size within the dust deposit will help us improve our mechanistic understanding of resuspension, as this is an important parameter in the material balance model, the size resolved surface concentration of dust particles. Another thing related to this is looking at how we collect the dust. So we may have very fragile agglomerates of particles within the dust deposit itself, and the various methods we use to collect the dust, either with a vacuum or a filter cassette, that may break apart some of these agglomerates and then shift the size distribution. So I think there's a need for kind of improving dust collection methods to better understand the size distribution of these particles. on all indoor surfaces, carpet, mattresses, ventilation ducts. Along with the size distribution of dust particles, we have the structure of the dust deposit itself. And this is something that I think is very, very complex, and also something we know very little about. These deposits can have varying porosity, as I show here. They can be very compact or fluffy. This porosity and also the agglomeration of particles within the deposit may depend on deposition mechanisms, compression of carpet or fabric fibers over time. So there may be a shift in this porosity and agglomeration of particles. We can broadly classify these deposits as heavy multi-layer deposits, where you have significant particle interaction, and then sparse monolayers where the particles are not in contact with one another. And this has been shown to significantly impact the number of particles that resuspend from a surface. In addition to looking at the deposit as in its entirety, we have to look at the individual particles that make up this deposit. And in regard at least to biological particles, we know that they are very diverse in their shape, their morphology. They're certainly not spherical, and they're not perfect spheres. They have very irregular surface features, and these are parameters that would affect their adhesion to surfaces as well as to each other. We know that these biological particles in settled dust can vary in size from less than a micron to tens of microns. For example, we may have single bacterial cells in the dust, as well as agglomerates of these cells. These may be 3 to 5 microns, whereas a single cell could be about a micron in size. We may also have fungal spores and pollen grains, as well as fragments of those, which can be much smaller, for example, on the order of 500 nanometers or so. And then we may have abiotic material, mineral dust or something of that nature, where if bacterial cells or fungal fragments attach to the surface. So there's certainly a lot of area here to connect microbiology and aerosol physics and improve our understanding of the nature of biological material in settled dust deposits. Last thing I'd like to discuss related to dust is adhesion. So we know there's a large body of literature on particle adhesion to surfaces, but this is narrowly focused on spherical particles on flat surfaces. And that's very much not representative of what we have indoors. We have non-spherical particles and very complex surfaces. For example, fabric fibers for clothing, bedding materials, and so forth. Carpet fibers as well. In particular, I think there's very little data, if any data at all, on particle adhesion to fabric fibers. And I think this is something particularly interesting. So for example, your shirt or your bed sheet, this is a very loosely bound fabric, so it's uniquely different from, let's say, carpet fibers. These fabrics are in very close contact with our body, an intimate contact, so they may accumulate moisture, skin lipids, and this accumulation of this stuff may affect particle adhesion over time. These particles may be embedded within these fibers. That's something we know very little about. How do these fibers vibrate when they're exposed to movements of the human body or airflow? So moving on, we'll now discuss resuspension and resuspension associated with different human activities. So the first, and I think most of us know, is that we resuspend particles when we walk. I think this is the particular activity that we know the most about, or it's the most well characterized. In this case, studies by Andrea Farrow's group at Clarkson looked at, using a mechanical foot in this case, resuspension fractions for different types of flooring materials, finding for at least for particles greater than a micron that we see more particles come off of carpet than we do hard flooring. There's also influences associated with relative humidity and so forth. But in addition to walking, we of course do other movements throughout the day, one of which is moving on a mattress. So this is a study I did at Texas with Mikael Spilak from Denmark, who's now at Drexel University, where we did a controlled chamber study looking at human-induced resuspension of mattress dust particles. In this case, we had a twin-size mattress that we artificially seeded with test dust and had volunteers, one of which was Rich, come into the chamber and perform a standard routine of 5 movements over 12.5 minutes. Here we looked at a variety of parameters such as dust load, ventilation rate, and movement type, and the intensity of that movement. But we found that these movements certainly do resuspend particles. This is area, again, we know generally very little about indoor air quality in these sleep microenvironments and exposure to particles or gaseous species while sleeping. But certainly we see that when we begin to move, we stir up dust, and the concentrations remain elevated throughout the duration of the movement period. Interestingly, when we stop moving, the concentrations may still remain elevated, so you may be exposed to particles that you resuspend as you fall asleep and lay still in bed. So the exposure period here might be beyond the period of movement. Here we also characterize the resuspension rate. We found, of course, of other studies that resuspension increases with particle size, and this is generally because the magnitude of removal forces, such as aerodynamic lift and drag, surface vibrations increase with particle size. Interestingly, we found that the body mass of the volunteers had very little impact on the resuspension rate. What really mattered in this case was not the body mass, but the intensity of the movement, how the people moved. So we asked the volunteers to perform 5 different movements, and we found that the more intense the movement was, as characterized by surface vibrations, the more particles you resuspend. So you can think that a full rotation of your torso would resuspend more particles than simply sitting on the mattress. So in addition to sleeping, we know that particles can detach from clothing. So this was a nice set of studies by McDonough and Byrne where they looked at particle detachment from this clean room outfit with different fabrics attached due to different types of physical activity. In this case, she was dancing this Irish dance. But certainly we see about a quarter of these particles that were initially deposited on the surface are resuspended. This is something I think very, very interesting, and another area where we know generally very little as it relates to just occupational exposures or just general clothing that we wear every day. Okay, so enough about adults. I think one area that's very important and not very well understood is the infant microenvironment, this near-floor microenvironment, and particle resuspension induced by this crawling activity. So this is a study that I did in the past 1.5 years working in Finland. These measurements were done at the Finnish Institute of Occupational Health in a very nice large-scale chamber there. We also worked with microbiologists in Kuopio, Martin Tabel. Here we used I'll just call it a simplified mechanical crawling infant. It's by no means perfectly realistic, but it does do this, what I would call modified belly crawl. So it does simulate kind of the impact of the hands with the carpet and, let's say, abrasion of the belly as it slides along the carpet. We sampled in the breathing zone with an optical particle sizer, moving along on this mobile trolley, as well as the bulk air. And here I think what was interesting is that we took the carpets. So in Finland, people don't typically have area carpets, or they don't have wall-to-wall carpets, they have area carpets, so we could take these, carefully fold them, and bring them to the lab. In this case, this allows us to look at real dust and real dust deposits, not artificial test dust, which I think is quite interesting. And we also collected the particles on an inhalable sampler for offline analysis of qPCR and next generation sequencing. So here's a video, I don't know if, okay. You can see it go along. And you'll see here in a second, I would say what I call resuspension in real time. And again, this is real dust from a carpet that is not excessively dirty by any means. These dust loads were about average, I would say, 1 to 15 grams per square meter. But you can see that the particles certainly do resuspend as this little guy crawls. And what we find is that the beginning of the crawling, you see a big burst of particles. This is the number, well, the mass and the number distribution in the infant breathing zone in real time, falling along on this mobile trolley. So you certainly see you spend a lot of particles down to 0.5 microns in size, and as large as 10, which is the upper limit of this particular instrument. And after the infant stops crawling, the particles remain airborne for some period of time as they deposit or they're removed via ventilation. In addition to the real-time data with the OPS, we have the qPCR data. This is data analyzed by Martin, Pavel, and Quopio. And here we can see variation. This is the dust, so cell equivalents per square meter of carpet. And then we have cell equivalents per cubic meter of air in the infant breathing zone. But first, I think it's interesting just to see how much variation there is for these various microbial groups, gram-positive, negative bacteria, fungi, and so forth, just among the carpets. And these are just among residents from Helsinki, Finland. And not only that, but for a given concentration in the carpet dust, there's quite wide variability in what gets into the air. And this could be due to a number of reasons, but I think this is certainly something very interesting where we need more focus in characterizing this kind of cloud of particles and microbes around the infants as they crawl. Moving on, so that data, this qPCR data is very nice, but it's offline, so you have to collect onto a filter for maybe a few hours or even longer to collect enough microbial mass to be able to detect the DNA. I think another useful technique, this is done by Bill Nazaroff, looking at laser-induced fluorescence-based measurement techniques to characterize bioaerosol concentrations and size distributions in real time. In this case, it was an occupied classroom. But this particular instrument, it will tell you if the particle fluoresces or not, which could give you an idea if it's biological or not, or an abiotic particle of some bacterial cells attached to the surface. But we can apply this technique also to resuspension, which I think is a very much an inherently transient process. It's by no means a steady process. We're moving, we stop moving, and so forth. The intensity of our movements changes throughout the day. Here we used one particular instrument developed at Tampere University of Technology called the BioScout, sampling in the breathing zone of the infant, as well as the adult breathing zone. This was for walking on the same carpet, so we could compare the concentrations of these fluorescent particles for both crawling and walking. And this is just a quick example, but we see that certainly when you begin crawling, you resuspend these fluorescent particles. There's like almost a burst of particles into the air. So you can see it's very much a transient process. If the baby was crawling, if they stopped crawling, we'd see changes in the size distribution and the concentrations of these particles, and the same for walking. So that was just total fluorescent particles. We can go a bit further and use some of the information from these instruments, such as the intensity of fluorescence of the particle, different fluorescence channels, and optical diameters and shape, symmetry factor, to classify the fluorescent particles in real time as either pollen, bacteria, or fungi. So this is just a paper that was just published by Mark Hernandez using a WIBS instrument, wideband integrated bioaerosol sensor. So I think this is something to be very interesting for resuspension and seeing how many bacteria you resuspend in real time, fungi, pollen, and then compare this with other indoor bioaerosol sources as well as outdoor bioaerosols coming into the building through ventilation and infiltration. Related to this, transient behavior is looking at the attachment process of particles in real time. This is at a very, very micro scale, but looking at each individual particle and the trajectory it follows as it comes off a surface. We may have direct liftoff, bouncing, rolling, and so forth, but certainly we can use this analysis, these high-speed imaging methods, for real indoor particles. These are spears. Of course, we don't have we don't have perfect spears in indoor dust. We have very complex particles in regard to their shape, as well as surfaces. So this would be something quite interesting, I believe. And lastly, I'm running out of time, is the air to breathing zone. So not just looking at the resuspension, but the transport to the breathing zone. One thing, as Lynn pointed out, is the source proximity effect. I think this is very much relevant for resuspension. This was shown with this Piper, this pre-toddler inhalable particulate environmental robotic sampler, where they sampled near the floor and showed that the concentrations of particles expressed as a mass concentration are much greater near the floor than they are at some stationary monitoring site, which is typically used in indoor field measurements. We saw the same thing in the infant resuspension study, showing that the breathing zone concentration is quite a bit greater than the bulk air, and the same applied to the qPCR analysis, where we saw that the ratio of the breathing zone in the infant breathing zone to the bulk air concentration is from 5 to greater than 10. So quite significant variation in regard to height. We also see this is a modeling study by Lindsay Marr's group looking at the vertical variation in resuspended viral containing particles. And we see that shorter people can be exposed to more of these particles than taller people. So the same concept applies here. And just to finish up, I think in regard to resuspension, it's something that we need to look at in regard to different scales. So very, very controlled small scale wind tunnel or chamber studies where you can look mechanistically at different parameters and how they affect resuspension. And then these full scale chamber studies where we have them for the walking, crawling, and sleeping are very nice because you can control the background concentrations and so forth. But then, of course, linking that with full scale studies in offices and homes. Really have a full holistic picture of particle resuspension. So I think I'll just end it here with Pigpen. So I think this is just a very short, brief overview of resuspension, but I think it's a very fascinating area of research, particularly as it relates to bioaerosols. And I think there's still a lot left to be learned, and hopefully we can do that in the coming years. I'd just like to thank my collaborators, some of which are in this room, with whom I worked on various projects related to resuspension over the past few years. So, thank you.
Thank you, Brendan.